I wanted to apologize to all of you. We had a major Zoom disaster today, so we don't know why the original Zoom link didn't work. That's why we had to send out a new link at the last minute. We sincerely apologize for the delay, and we thank our speaker and all our audience for their patience. So before I introduce our speaker, I had a few logistic things to talk about. This CME activity is jointly provided by the Queen's Medical Center and the University of Hawaii Jabson Department of Geriatric Medicine. As a reminder, evaluations are required for CMEs. You can find the evaluation form by following the link in the chat at the end of this presentation or on our email invitation or on our website. All speakers do have to give a conflict of interest disclosure. Dr. Rebecca Edelmayer has declared her conflicts and they are on her slides. Essentially, she's a full-time employee of the Alzheimer's Association. The Alzheimer's Association does forge partnerships with various other organizations around the globe, the NIH, universities, corporations, and the pharmaceutical, biotech, and device industries. So they're very transparent that they received 1.66% of their total 2024 contributed revenue from biotech, pharmaceutical, diagnostic, research industries. That's her only conflict of interest. So it's my great pleasure to introduce Dr. Rebecca Edelmayer. She's a distinguished neuroscientist and pharmacologist, currently serving as Vice President of Scientific Engagement at the Alzheimer's Association. She earned her PhD in medical pharmacology from the University of Arizona and a bachelor's degree in neuroscience from the University of Pittsburgh. Her career spans significant contributions across nonprofit, academic, and industry sectors, including senior scientific roles at AbbVie and Abbott. With extensive experience as a scientist and educator, she unites researchers and clinicians on topics related to neurodegenerative diseases, biomarker testing, drug discovery, clinical trials, and real-world evidence studies. Her leadership continues to drive advancements in dementia research and treatment, making a profound impact on the field. At the Alzheimer's Association, she leads several national and international initiatives in dementia science. She's a study investigator for the Alzheimer's Network for Treatment and Diagnosis, or ALZNet, a real-world data initiative for new FDA-approved Alzheimer therapies. She also oversees the AAIS to advance Alzheimer research and treatment, called the iStart, an inclusive global network of over 13,000 researchers, clinicians, and dementia professionals. Her team continues to expand the association's suite of global scientific meetings where theories, breakthroughs, and best practices can be shared. She serves on steering committees and advisory boards for cutting-edge scientific consortia established by the NIA, including the AMP-AD, Model AD, and Treat AD. She's dedicated to advancing Alzheimer's Association's scientific agenda through research, education, and community engagement. Please welcome me in joining Dr. Edelmayer.
Thank you so much for the introduction and really kindly thank you all for being able to join this afternoon. I think I want to be able to take you really on a whirlwind tour, I should say, of some of the things that are really happening in our treatment and disease space. One of the things that I had been asked to focus on today was really on the diagnosis and staging of Alzheimer's, as well as some treatment. As noted, my disclosures were already noted, but you can find more information on our website too about our organizational disclosures if that's helpful. So I thought what I would do today in terms of our time is I think we'll start off with some principles that will really aim to go over some of the new revised criteria for diagnosis and staging of Alzheimer's disease. And then I want to touch on the landscape of tools for early detection and diagnosis and some of the tools that are being developed for you as clinicians. Then we'll touch a little bit on the latest advancements and treatments that are in the pipeline and focus a bit more on real-world data and real-world evidence, which is where our field is moving towards. So just to set the stage today for you all today, Many of you may be familiar with the Alzheimer's Association Alzheimer's Disease Facts and Figures report. This comes out every year. And we actually are about to launch our 2025 report in just a couple weeks. So I encourage you to check back in on some of these numbers that I'm about to show you today, because they will change over time as our report continues to grow over the years. But you know that Alzheimer's disease and other types of dementia are impacting your community in a very significant way, not only for those that are living with Alzheimer's disease and other types of dementia, but also for their caregivers. And we need to continue to do the research that helps to provide better routes for us to continue to diagnose and detect these diseases, understand the prevalence and risk, and understand how we're going to treat and care for individuals that are living with these diseases. As you may know, that the number of Americans that are living with Alzheimer's disease is really expected to double by 2060. And you can see that this continues to grow year over year, mostly driven by the demographics of our aging population. We know that age is one of the biggest risk factors for Alzheimer's disease, but we know that there are other factors that drive risk. And certainly we have learned that there are some disparities that exist within Alzheimer's disease and other types of dementia. We know that almost 2/3 of Americans that are living with Alzheimer's are actually women. We know that older Blacks, African Americans, Hispanics, Latinos are more disproportionately more likely than non-Hispanic whites to have Alzheimer's or other dementia. But we also know that we need to continue to drive forward more population-based studies for other racial and ethnic groups. We do not have very, I would say, significant data across all populations to be able to give some of those demographic and prevalence numbers. And what may drive impact or risk of cognitive decline is really important that we continue to study this. We know there's both non-modifiable risk factors, but also modifiable risk factors that drive risk. And it's this really constellation of reasons that may be both fundamental as well as unique to everyone that we have to continue to study. And the social determinants of health, things that your patients might be experiencing across their life course, may impact some or even all of these factors for them. And we need to continue to study all of these factors because the strength of our understanding about each one of these is different. In particular, you can look at things like cardiovascular health, where we have a much stronger evidence base in terms of what may drive risk intervention versus something like traumatic brain injury, where we're still learning more. So we need to continue to study this from all angles. So I just wanted to keep this all in mind as we moved through the discussion today, because I'm not going to focus a lot on the basics of Alzheimer's disease and other dementia, but really focus on some of the really guidance and around diagnosis and staging for Alzheimer's. And this is really based on probably over 120 years of work that's been happening in the field. When the disease was first identified in 1906, when we're speaking about Alzheimer's disease specifically, by Dr. Alois Alzheimer, who did this with his team back over 100 years ago. But when we started talking about how to actually identify and diagnose Alzheimer's, a lot of this started with guidance that was put out originally by the NIN-CDS ADRDA, which the ADRDA is actually the Alzheimer's Association, back in the day, working through a task force that helped really identify not only standardizing Alzheimer's disease diagnosis based mostly on the clinical symptoms back in that day. And then in 2011, there was a publication of new diagnostic recommendations by the National Institute on Aging and the Alzheimer's Association for the diagnosis and staging of the Alzheimer's disease as really a continuum that begins in preclinical stages or asymptomatic stages, moving through mild cognitive impairment and then dementia stages. And we were introducing the concept of Alzheimer's disease as both a clinical but also a biomarker construct at this time, and biomarkers that initially included cerebral spinal fluid and imaging. And it wasn't until 2018 where we started seeing more publications that were really focused on talking about the ATN, or what we call the ATN framework. This was really a research framework that was published to formally introduce Alzheimer's disease as a biological construct defined by both amyloid, tau, and neurodegeneration biomarkers across that disease continuum. And then it was in 2024 that we recently published a revised criteria for the diagnosis and staging of Alzheimer's disease through an Alzheimer's Association work group. And this will then now go on to be developed into an actual clinical guidance from this this revised criteria. But I want to talk today a little bit more about what that criteria looks like. So the conceptual foundation from the prior guidances that I just went through, it was identified that it was really necessary to separate the syndrome, the clinically identified impairment, from the biology, some of the hallmark changes that are occurring in the brain. We know that Alzheimer's disease is defined as a biological process that really is detected by these core biomarkers throughout the disease course. And we know this continuum begins with the appearance of neuropathy and some of these biomarker changes, while people are still very much asymptomatic, and progresses through stages of increasing pathologic burden and eventually leads to the appearance and progression of clinical symptoms. And so when we thought about the developments that have also occurred since the 2018 ATN research framework, this really prompted the update for our 2024 criteria. We saw a lot of changes that were happening in the space. Certainly the development of plasma biomarkers, some or not all that actually have very excellent diagnostic performance, even equivalent to approved cerebrospinal fluid assays. But we also saw that there were approved treatments that target the core disease pathology. And it was really important that we started to bring the field together around the conceptual alignment, not only across industry, academia, and clinicians around biomarker classification, diagnosis, and staging of the disease. So we published in March of 2024 the revised criteria for diagnosis and staging of Alzheimer's. This was a work group that included key experts from across both the clinical and research spaces, and it was really brought together objective criteria for the diagnosis and staging of Alzheimer's that incorporate these recent advances in biomarkers, but also create an integrated biological and clinical staging scheme to really serve as this bridge between research and clinical care. And you'll see as I move through this, the biological staging scheme is actually in letters, and the clinical staging scheme is really in numbers that really brings together also the draft guidance that the FDA has actually put forth and incorporates this all into one particular criteria. When we looked at the criteria for diagnosis and staging of AD, this was really meant to be intended to provide general principles to inform diagnosis and staging of Alzheimer's that reflect the current science. It's not a specific clinical practice guideline. As I noted, we will actually be building that now, and we've already started the process of putting together that expert panel who will come together to bring the recommendations forward. The clinical use of the Alzheimer's disease biomarkers is really presently intended for the evaluation of symptomatic individuals. It is not intended to be used for cognitively unimpaired individuals. And the way they put this guidance together was they looked at developing sort of a categorization of fluid analyte as well as imaging biomarkers. And it brings together this idea that there's both core 1 and core 2 biomarkers that can be distinguished by their timing of onset in terms of the changes that are happening in the brain, as well as their intended use in an individual. The core 1 biomarkers incorporate the amyloid and the T1 biomarkers, which can be phosphorylated and secreted Alzheimer's disease tau. So you can see some of these biomarkers include both imaging as well as fluid biomarkers. And then in core 2 biomarkers, we're looking at tau proteinopathy, which can be fluid biomarkers such as the MTBR tau 243 or other phosphorylated tau forms that would be much more focused on the tau progression or tau PET. And then you can see there's other biomarkers that are included as well and noted in this criteria, but these are not necessarily considered core biomarkers. Those that help to define neuronal injury, those that help to define inflammation, and even vascular changes and synuclein changes that are not part of this core categorization. But I'll come back to this in just a second, because what this is really trying to represent is that when you look at the core 1 biomarkers, these are really representative of sort of the early biomarker changes that may be occurring and then are sustained or remain throughout the disease process. And the core 2 biomarkers really are a little bit more representative of the tau processes that are occurring in the brain. And as you can see, both of these, at least the core 1 biomarkers, tend to align temporally with the early changes that are happening in the disease. And then the tau biomarkers tend to align a bit more with the changes that might be seen clinically. But all of them are changing over time in terms of their biomarker abnormality. When we look at the diagnosis of Alzheimer's disease, it can actually be established by abnormality on specific core 1 biomarkers like the amyloid PET or cerebral spinal fluid changes that may be occurring in both the amyloid markers, but also ratios of CSF p-tau 181 and amyloid beta 42, or a cerebral spinal fluid test that's looking at total tau and A beta 42. And there are some very accurate assays that could be used as a single assay or even in combination as a multi-analyte or ratio process. The core core 1 biomarkers can be used for the early detection of AD in people without symptoms, and also the confirmation of AD in an underlying pathology in someone with symptoms. And the core 2 biomarkers are really not typically standalone tests for the diagnosis of Alzheimer's disease, but can be used in combination with core 1 to really stage the biological disease severity. And that brings me to kind of the biological staging process when we're talking about these biomarkers. So biological staging, which is the biomarkers versus the clinical staging of assessment, staging can really be accomplished by, 1, a combination of amyloid PET and tau PET, or 2, a combination of core 1 fluid biomarkers, which would really establish a biological stage or higher, plus some of this tau PET, which could be used to discriminate among some of the stages. And this is an example of a description of early or initial, early, or intermediate and advanced stages of PET that would put an individual in either the early stage biomarker versus all the way through the continuum of advanced stages of biomarkers. And biological staging can really only be completed by using sort of these core biomarkers. We're not using any of the additional biomarkers like vascular biomarkers, inflammation, or neurofilament light types of markers that would be used for staging purposes. And this is then combined with clinical staging for individuals on the AD continuum. So when we look across the different stages that exist, and this again goes back to sort of what has been proposed by the FDA and their framework for clinical staging, all the way from stage 0, which is an asymptomatic individual that may also contain a deterministic gene, all the way through stage 6, which would be dementia with severe functional impairment. And so you might really look at this then from the disease continuum of asymptomatic all the way through stages of mild cognitive impairment, all the way through mild, moderate, and severe stages of dementia. And when we look at this integrated biological, then, which is the letters, and the clinical, which is the numbers staging, you get something that looks like this chart that actually comes out of the publication, which looks at this integrated level of staging. One of the things to note here is, why are we looking at these sort of orthogonal staging axes? And what we've learned from Alzheimer's disease and the sort of staging and the criteria and the work group has suggested is that, there can be individuals that don't fall along sort of the general expectation of what we would see in terms of disease continuum, disease trajectory, and even the speed and severity of not only their biological changes, but also the symptom changes that may happen over time. So because the biological and clinical stages don't always track in lockstep for all people, this is why we need to have this sort of staging in this manner. But what you're sort of seeing along, I think, this diagonal line is really noting that this is more of like a expected or typical trajectory for some individual, but they may fall on either side of this line. One thing I do want to note to all of you is that what we're seeing in Alzheimer's disease is not atypical in the sense that we're seeing this type of integrated biological and clinical classification system that's happening across other neurodegenerative diseases. And we do have tools, even in other neurodegenerative diseases, to start transitioning to this integrated system, both for Huntington's disease, Parkinson's disease, and other synuclein diseases. And so when we look across all of these different diseases, we can see and suggest that neurodegenerative diseases can exist with both a clinical diagnosis as well as a biological staging framework. There's common elements in modern criteria across all of these neurodegenerative diseases for Alzheimer's, Parkinson's, dementia with Lewy bodies, and Huntington's disease. There's the biological definition and diagnosis. This really lacks a specificity of clinical symptoms, but does enable disease-specific treatments. But an integrated staging uses both biology and symptoms. And that allows us to enable matching both the optimal treatment, but also the stage of an individual to each treatment. And we can think about this a little bit more like a precision medicine or a personalized medicine approach, like we do in oncology today. And we need to really be considering the disease continuum. So preclinical treatment eventually is the goal. We do want to be able to catch individuals at the earliest stages of disease before the extensive and irreversible damage that might become present in an individual is allowed to set in the brain. And there is the potential, and you'll see some data from clinical trials that I'm going to demonstrate in just a few minutes about the potential to be able to treat or even prevent slowing some of that damage. So overall, a quick summary of what has come out of the revised criteria for diagnosis and staging of Alzheimer's disease is that we really are focused now on diagnosis by core 1 biomarkers, and that you can stage the disease itself by using both core 1 and core 2 biomarkers. And that if you use this integrated staging of the core biomarkers and the clinical symptoms, it allows you to have a more holistic picture of what may be happening in a particular individual. And really, the conceptual as well as the operational diagnostic and staging alignment across not only Alzheimer's disease can occur across a spectrum of neurodegenerative diseases like Huntington's disease and synucleinopathies. So I want to touch base quickly with you on some of the progress we're seeing in tools for early detection and diagnosis, because there's a lot more work we need to do to be able to move us into a full guidance for diagnosis and staging of Alzheimer's disease. And some of that incorporates other types of clinical practice guidance. And I wanted to make you aware of other things that are in the works that the Alzheimer's Association is helping to lead, which is one is a clinical practice guideline that's in development for cognitive assessment of tools for cognitive impairment. And this is really designed to be a clinical practice guideline on the use of assessment tools for detection in primary care settings. So the types of tools that could really be used in detection, not necessarily diagnosis, but really trying to capture people at some of those earliest stages, but that would then need additional confirmation through either additional biomarker evaluation and potentially additional neuropsychological testing. This is CPG is in development today. We do hope to actually have this published this summer. You will see an opportunity for open public comment soon as we continue to pull the evidence together and the recommendations are created by the panel. And then this will be built out into different types of clinical tools, as well as educational courses and presentations. And we're using sort of the state-of-the-art grade methodology that helps the expert panel to really move from the evidence that's been published into decisions or recommendations to help provide those clinical practice guidelines. In addition to that, we're really, really focused on following all of the biomarker tools and technologies that have been in development. And you can see, similar to how we've updated our clinical guidance over time, where we first started in 1906 with autopsy tissue and really doing immunohistochemistry to identify some of the pathology. We've moved into other types of technologies, cerebrospinal fluid, you've heard me mention amyloid PET, tau PET imaging, and we're really now in an era of new diagnostics that incorporate plasma testing as part of the analysis or part of the workup for patients. And we're seeing these advancements happen really, really rapidly. And there's a lot of work happening in blood-based biomarkers. It's a very, very active area of research. We know there's a number of tests that are not only on the market today that are available for clinical use, but there's a number that are still actually in a sort of, or what would be considered research use only. That are still in development. And there's a lot of progress being made. There's not a single FDA-approved blood biomarker test yet today to help diagnosis or aid in the diagnosis of Alzheimer's disease, but we know there are some that have been submitted to the FDA for review, and we know that there's a lot more in the works for the future. But one of the things that this really kind of lays out is that not every test is created equally. And while we did publish the revised criteria for diagnosis and staging of Alzheimer's disease in 2024, it did leave open the opportunity for us to continue to develop more revised guidance to clinicians on how to use some of these blood tests and plasma tests that are being developed today. And really, the accuracy of some of the emerging blood tests needs to be considered as we try to guide clinicians to the use of these tests in aiding in their clinical decision making. So we needed to create a formal guidance, a formal clinical guidance on biofluid-based biomarkers that helps to really distill the best available evidence and translate it into clear and actionable recommendations for clinical practice. And this is just to share with you that there is an ongoing clinical practice guideline in development for blood biomarkers for Alzheimer's disease. And we do hope to have this also published by our Alzheimer's Association International Conference this summer in July. And this is really focused at aiming some of those initial questions. And in terms of the initial population, we will be creating a guidance that helps to provide recommendations for clinicians who are in specialty care practice, who are seeing individuals with cognitive impairment, and whether or not these tests can be used as both confirmatory or triage tests within a clinical practice. And the clinical practice guideline will actually be rapidly updated to go on to making recommendations about the use of these tools in individuals in specialty care that are cognitively unimpaired. And then we will be moving on to primary care and the recommendations for primary care clinicians on the use of these tools and technologies within their clinical setting. So a lot of work really happening in not only translating these biomarker results for patients, but also helping to make sure that we have tools at your fingertips to help support the work that you may be doing in the diagnosis, treatment, and care of individuals with Alzheimer's disease and other types of dementia. Because we know that you're being asked many, many questions, really trying to understand and help support the patients that you treat in terms of the questions they ask around their disease stage, a diagnosis, if they're at risk, if we even have tests and tools that can help support that type of answer for individuals. We have to be able to look into the evidence that exists today to make sure that you have really the right test for the right patient at the right time. And all of this is so, so important at a time where we know that you are being bombarded by information too around a lot of the new treatments that might actually be available for patients. So I did just want to quickly touch on some of and then give you a brief history of what's happening right now in the Alzheimer's disease space, where we've been following quite closely the FDA-approved therapies for Alzheimer's that have been designed for a variety of different usages. Some are really designed to treat symptoms of cognition and function. Those were some of your first generation therapeutics to help treat individuals with mild dementia all the way through severe dementia. We also have treatments that are able to help support very challenging symptoms like mood and behavior, things like sleep disturbances or even agitation for individuals that are living with Alzheimer's disease. And we have some of our newest treatments that have been approved by the FDA to slow disease progression. And the two in purple that you see there are the two newest I would say molecular specific targeting therapies. They go after the amyloid beta in the brain that is one of the hallmark disease pathologies associated with Alzheimer's disease. And again, just to give a little bit of an overview on some of these newest treatments, because they are, I think, causing the biggest change, I think, for our field at this time, because it really it necessitates that clinicians are able to actually identify the appropriate patient population, the appropriate stage of disease for these treatments, individuals living with mild cognitive impairment due to Alzheimer's disease or mild Alzheimer's dementia, as well as the evidence of the buildup of amyloid plaques in the brain. And we need to do the biomarker testing to be able to support some of that. And there's a variety of different tools that are out there that could be helpful to support clinical decision making using some of these newest treatments. There is a tool I want to make you aware of, appropriate use recommendations that were published in 2023 that helped not only discuss the treatments themselves, dosing, administration, but also the importance of monitoring for side effects for these newest treatments, especially for something such as ARIA, which is the amyloid-related imaging abnormalities. That is one of the side effects of these treatments. There is also appropriate use recommendations for donanemab as well. And with this, again, this was just published actually just a few weeks ago that came out by the clinical expert group that put together these recommendations. And just to briefly go over with you some of these newest treatments, this is information directly from the USPI that really tries to draw together, because we get a lot of questions about the differences between these two treatments. And they are both designed to target beta amyloid, but in terms of the mechanism of action of how they actually help to remove the amyloid from the brain, it's slightly different. So you will also have some slight differences in terms of the efficacy as well as the safety of these administering these therapeutics. But one thing to note is that these are not head-to-head comparisons. This is really drawn straight from the prescribing information and the labels. And you must note that the information comes from two wildly different study designs when looking at actually testing these treatments in the phase 3 clinical trials. So just to give you at least a better eye view of what this looks like in graphical format, you can see that when we look at some of the efficacy data from both lecanemab and donanemab, when we look at the CDR sum of boxes, which was the test used as one of the primary outcomes for these clinical trials, you can see that they're able to reduce or slow the cognitive decline that's occurring when compared to placebo. And I think just flipping back to the USPI, you can see that this is in the range of between 27 to 29%. So fairly similar across the treatments when you look at both the CDR sum of boxes, the ADS-Cog test, as well as the ADCS test that was used for these clinical trials. But then I want to also demonstrate to you some of the safety. And this is a comparison of one of the side effects that occurs with the amyloid targeting treatments, these amyloid-related imaging abnormalities, which are monitored through regular MRIs. You can see that the overall ARIA rate across these two treatments is slightly different compared to placebo. When you look at treatment with lecanemab, around 21%, and donanemab, 36%. But I think when you look across the board, one thing is important to note is that when having conversations with patients about the potential benefits, there also need to be conversations about the potential risks of these treatments. One thing that's really interesting that we continue to follow is that there's more and more data coming out every day about these new treatments as they roll out onto the market into broader populations, as they continue to also follow these patients in open label extensions. And I just want to give you an example of data that were presented last November. And then I've also added to this something that was just presented at the Alzheimer's disease and Parkinson's disease conference just a couple weeks ago that happened in Vienna. They presented in November 6-month data from the Trailblazer All-6 study, which was demonstrating that for donanemab specifically, when they ran a number of different dosing regimens as part of this Trailblazer All-6 study, they were actually looking to modify the treatment dosing to see if it would have an effect on ARIA. And based on a 24-week data initially, they were able to see a change in the initiation of the donanemab dosing by shifting one vial from the first infusion to the third infusion, which resulted in significantly lower ARIA-E and significantly lower symptomatic ARIA-E. So being able to just titrate or modify the start of the medication with a lower dose is able to reduce ARIA-E and symptomatic ARIA-E across even ApoE4 genotypes in this modified titration arm. So this really continues to demonstrate to us an example of why we need to continue to gather data after initial approvals have happened. And we're continuing to see more and more research presented around the safety of both donanemab and lecanemab, not only through their clinical trial data, but we hope to see more presented out of real-world data that's also continuing to be gathered. So this brings us really to a new phase of treatment. We started seeing some of these big changes for our field in 2021 with the first approval of aducanumab, which was the first amyloid targeting therapeutic that has now been discontinued as of last November of 2024. And now with the approvals of lecanemab and donanemab, but this is not where we're stopping. And I want to be really clear in this talk. I've focused because we're getting a lot of questions on these newest treatments, but there are a large amount of treatments in the clinical trial pipeline today, over 120 unique therapies that are being tested in clinical trials that target multiple aspects of Alzheimer's disease biology. Not just targeting amyloid beta like we've been discussing, but there are many, I would say, phase 2 clinical trials mainly. I think we're starting to move into phase 3 clinical trials on tau. We've seen other trials that are testing drugs, I would say, that would target like the metabolism or the cellular energy systems, like the bioenergetic systems of the cells. We've seen disease targets that are looking at neuroinflammation and the immune system. And this might give you a bit of a better perspective of all of the different therapies that are being tested today. This is actually comes out of Dr. Jeffrey Cummings and his team that does an annual report in the TRCI, Alzheimer's and Dementia Journal. We expect that there will actually be a new report coming out within the next month or so that gives us the 2025 update on the Alzheimer's drug development pipeline. And you can see in this pipeline from 2024 that there's over 127 unique drugs that are being tested across 164 clinical trials. One thing that's important to note is that some are these what we might call disease-modifying therapeutics or disease-modifying treatments that really aim to slow down the disease process. But there are also treatments that are symptomatic treatments, ones that are aimed at being cognitive enhancing or even helping to address some of the behavioral symptoms, agitation, aggression, psychosis. And one thing to note here is there's about 31% in the clinical trial pipeline that are considered repurposed agents, agents that may be coming in from other different disease or therapeutic areas like cancer, like immunology. And it's really critical that we continue to follow this. We continue to see over and over, year after year, that continued investment, continued research is driving, I think, massive changes across the diversification of this clinical trial pipeline. Because I think we really truly believe that it is likely to be a combination approach of treatments, really powerful, effective therapeutics working together to target the disease biology of Alzheimer's disease, but also other types of diseases that cause dementia in the brain. I want to wrap up my talk today with really giving you some insight into kind of where the field is going. We are certainly watching that clinical trial pipeline diversify for Alzheimer's disease and all other types of dementia. I didn't even show you the trial pipelines for other types of neurodegenerative disease studies. But we're moving into a new era that actually is going to allow for the use of real-world data to inform treatment and care and really help to support clinical decision-making. And in 2021, the Alzheimer's Association really saw a need, but also a desire for the field to be able to continue to have an infrastructure and create an ecosystem that would allow us to collect additional real-world data from a broader setting of clinical I would say a broader, I would say, population of people seen in a variety of clinical settings. So we actually launched something called ALZNet. This is the Alzheimer's Network for Treatment and Diagnostics. And this is really a voluntary healthcare provider enrolled patient network that is enrolling clinical trials, or sorry, excuse me, clinical sites, not clinical trial sites, clinical sites all across the country. You actually have one in Hawaii, which we're very excited about, and we hope that more clinical sites will join. This is really a way for clinical sites to consent their patients to be able to collect data from them over time, collect clinical data, imaging data, diagnostic information, medical history, all kinds of side effect data as well that these patients would be being evaluated for or treated for some of these novel Alzheimer's disease therapies that have been approved in 2021 or after. So today, we currently have data from lecanemab, donanemab, aducanumab, as well as brexpiprazole, which is used to treat agitation associated with Alzheimer's disease. And this is not a clinical trial. It's really implemented in real-world clinical practice. And sites are the ones that are providing that routine care for individuals, diagnosis and treatment, and they're helping to support information and data upload into the ALZnet database. And it's really designed to work collaboratively with other studies that can use ALZnet as sort of that framework or even scaffold to build other studies on top of it. So even federal agencies or industry can use this, or academics can use ALZnet as a space to continue to perform research. And it'll allow us to really answer a lot of questions. Especially around what the long-term effectiveness of these treatments are, the long-term safety considerations, who's going to be a responder and not a responder, and who is going to potentially have treatment-related side effects, and which treatments are working well together in combination, and who really has access to treatment and who does not. We have to think about this from an equity standpoint, and how do we continue to make sure that we're following everyone that we can to support the most informed clinical guidance back out into the field for you as clinicians to be able to learn from in real time. So just to give you a snapshot of the kind of information that ALZnet is collecting, today we have over, I would say 1,700 patients that are enrolled. This number continues to jump every day. We have 95 clinical sites that are activated across the United States, and we hope to have more. These could be a variety of clinical settings. We have neurology practices, both private and public. We have large academic research centers like the Mayo Clinic, and I would say I'll give other examples like Brigham and Women's. We have University of California, San Francisco, but we have small geriatric practices as well. We have single, I would say, and group offices that are up and running, and we also have an imaging network that we've launched. So this would allow for imaging facilities to join, because we know imaging is not always conducted on site within a particular clinical setting. So this allows to really bring providers together across the nation. And we think there's a lot of benefits to participating. We ALZnet is actually approved by the Centers for Medicare and Medicaid as a pathway towards coverage of use of the anti-amyloid therapies if they've received traditional FDA approval. So it is one of the CMS approved registries if you need to register any patients to actually allow them to receive coverage for them. It allows us to continue to help support sites through the operation as well as the data collection for ALZnet. We do provide some site remuneration, and we really believe we've really designed ALZnet to be an ecosystem so that it helps to support clinicians for you to gain insights off of your patient populations through data sharing. We're going to be launching in July the ALZnet data dashboard, which would feed information back to you as a clinician and site about the patients that you are treating, so that you can see aggregated, de-identified data in a way that may show patterns in prescribing, patterns in side effects, patterns in diagnosis that could allow you to inform your clinical practice. And we really are trying to bring together a wealth of resources that allow you to have access to information, educational support, and really guidance to help you continue to do the amazing work that you're doing for patients really across the state. And so I hope today I was really able to give you sort of a broad, I think, overview of some of the things that are happening in diagnosis, staging, as well as treatment. And I really think we're in a new era of research, treatment, and care. When you think about, if you were to put Alzheimer's disease at the center of this pinwheel and think about what we've learned over time about the brain changes that are happening, the changes that are really driving the biomarker development, not only in fluid analysis, but imaging analysis and even other types of emerging markers, and how that actually supports therapeutic development, not only the diagnostic pipeline, but really how is it going to drive the right therapies to the right patients? And you can look, for example, at the two therapies that are listed here as really driving at a very specific part of that disease pathology that we know is associated with Alzheimer's disease. But we know there's a lot more that we still need to do to be able to tackle this disease holistically. And when you really think about it, and I didn't even get a chance to cover this today, there's a lot of work that's also ongoing that's studying risk factor interventions. How do we actually reduce risk from the onset? And could we even be doing lifestyle interventions even as a therapeutic strategy? And how do we combine these together to really provide the most sustainable strategies for patients moving forward? So I think overall, it's a very, very exciting time. I think we're starting to see really a paradigm shift in diagnosis and staging, not only of Alzheimer's disease, but other types of dementia that really look at this integrated staging. There's a number of new tools for detection and diagnosis, as well as clinical practice guidelines that are in development. And I really encourage you to keep your eye out for those that are coming out later this year. There's a number of new approved treatments and more in the pipeline. And we really have a lot of therapies that are under investigation. I think I really just demonstrated to you the Alzheimer's disease pipeline, but there are so many. I think there's probably, I would say a few hundred trials that are not only looking at treating Alzheimer's disease, but other types of dementia. So many more than that 127 that you're seeing today. And I think just to close out, it's the real world data and real world evidence that are our next frontier. It's really exciting to see that we're in an era right now that's allowing for this. And we've seen a lot of success in other disease areas like cancer, multiple sclerosis, and other diseases of the brain that have really seen success and value in continuing to collect real world data as a way to inform and improve upon clinical care. So we do definitely think it's a new era. I hope this was helpful for all of you. One thing I do want to leave you off with is we have our Alzheimer's Association International Conference that is happening in Toronto, Canada this year and online. So if any of you are interested in attending, I would highly encourage you to attend. There's an opportunity for CME as part of the AAIC conference. We are really trying to make sure that we have a lot of clinical education that's provided as part of AAIC. One of the things I should also note to you all is that we have the Developing Topics abstract submission that just opened today. So for the next 2 weeks, if you're interested in submitting an abstract to present either a poster or an oral presentation, it's your last chance to get some of those abstracts submitted before we close it finally for the meeting. But I really encourage you to think about participating, where you'll see even more updates about Alzheimer's and all other dementia research and treatment and care that are happening at this conference. So with that, I think I'll stop sharing and make sure I leave a little bit of time for a Q&A. I know I talked really fast and covered a lot of information, but I hope that was really helpful for you and the team.
Dr. Edelmayer, thank you so much for a wonderful presentation. Lots of information, I agree, but very, very interesting. Let's open it up for questions. We have not a very large group, so if you would like to unmute yourself and just ask your question, please go ahead and do so.
May I start with the question?
Of course, Abe, go ahead.
Dr. Edelmayer, thank you very much. That was a wonderful presentation. And it's really exciting to hear about the developments in the biomarker space. But you mentioned how the amyloid PET scan was developed in the 2000s, and we still do not have an amyloid PET scan capability in Hawaii. Queens is hoping to have that available soon for our population by the end of the year. And so there's been a lot of developments, but in terms of making that available to the patient population is somewhat limiting. And so what's really exciting is all the plasma blood testing that is in development. And I know they're on the market already. But without FDA approval, it's hard to get it approved by insurance, and it's hard to say exactly how reliable the results are. So I'm wondering if you have a crystal ball that gives us a sense of when we might start to see FDA approval for those tests.
These are really great questions. And so I think I have a mixture of answers for you. I think one of the things that we know is that at least In other disease areas, while FDA approval is important, we think for some of the insurance practices, the CLIA approval does allow for the use of many of these tests. I think the one thing that we're really focused on is making sure that the tests that are being used actually have the evidence behind them to designate whether they're actually accurate or not to be used for the specific purposes. And one of the things that the clinical practice guideline is very, very focused on right now is assessing all of the published criteria that would actually allow us to look at the specificity, the sensitivity, the prevalence of the population that was actually tested to not only understand the analytical validation of these tools, but also the clinical validation of these tools, and it's particularly in the population that they're used in. For doing that testing, because there's one thing to say a test has analytical validation, right? But can it actually be used to be predictive in a clinical setting? And that's different. And what we're seeing, I can tell you, is that there's a pattern where we're seeing even publications come out where they publish the data in some of these ROC curves, or the area under the curve kind of AUC curves that you see. They publish them in a way that doesn't actually help you as a clinician, because they're combining both cognitively impaired people and cognitively unimpaired people in the same analysis. But a patient doesn't walk into your office saying, I'm both cognitively impaired and cognitively unimpaired. So this is the kind of analysis that's very, very strategically being done right now by the biofluid-based biomarker cognitive CPG, excuse me. So what we're trying to do with that is hopefully at least provide you the guidance, even in absence of an FDA approval, on what good looks like. What should be the standard for test? What does accurate actually mean? Is that 90% accurate? And in what sort of clinical setting is that accurate? And can the test be used as a triage test, we would say, or does it and then need to have a confirmation, maybe with cerebrospinal fluid? So that guideline is really looking at what we would call the gold standard, PET, or cerebrospinal fluid, or pathology, to really truly understand whether or not that blood test is recommended to be used. And so I would say we hope that that's going to provide better guidance for you, because right now we know the revised criteria does fulfill, I think, some guidance around cerebrospinal fluid. So hopefully that will be very helpful to you in Hawaii, knowing that you're still waiting on your PET imaging capabilities. But it does sort of leave a little bit unknown around plasma testing. And it says that the test should be accurate, with at least as accurate as a cerebrospinal fluid test should be. And that's where we think this clinical practice guideline that is in development will be really helpful for that. If I were to read into my crystal ball about an FDA approved test, I would say that we know, and this is publicly available information, we know that there is one company, Fujiru Bio, that has submitted their test to the FDA, and it is currently under review, one of the plasma tests, but we know that there's likely more to come.
Excellent. Thank you for that. I look forward to the publication of your guideline.
Are there any other questions? I have one question, Rebecca. So these clinical practice guidelines, I think, will be very helpful for when we're seeing patients. Do you see that these guidelines are also going to be applicable to longitudinal epidemiologic studies of large populations?
In terms of being able to guide how you should collect the information, or are they pulling
No, in
data
terms
from?
of diagnostic capabilities, because most of the studies use CDR for staging and NINDS ADRDA criteria for clinical diagnosis. Do you see that these biomarkers and other imaging studies might replace that in larger studies as well?
I think what you're going to continue to see is that from the discussion that's been happening in the field, there really needs to be both a clinical evaluation and a biomarker evaluation for these diseases. I think that trying to answer your question very carefully because I know that this is continuing to evolve over time and how we will use the guidance. So the revised criteria today for diagnosis and staging is that very much. It's a criteria. It's not a clinical guidance yet, but it is working its way towards that. And I think that it can help to inform some of the information that could be gathered, in the future, even more from like a health services research perspective when it comes to epidemiology, so that we are collecting the right information from patients over time. But I think you'll see even some of the largest cohorts that we have today, things like the ADNI data sets, and some of these they're really collecting a variety of biomarkers over time. But I do think that the way that we've done guidelines in the past, I would say, in our disease space has not been, this is a very evolving and a new science. Clinical practice guidelines and using the grade methodology is relatively new. But it is the most rigorous. I'll have to say, as someone who's sitting on the panel for the biomarkers, it is one of the most rigorous things that I have ever seen. And what it does, though, is it does a lot of this work for all of the clinicians on the background to distill all the information and drive the evidence to decision by the panelists, so that when you actually come out with a recommendation, you can feel comfortable about the strength of that recommendation. If there's still limited evidence, the recommendation may be rather weak to use something, but that may change in 6 months when more publications come out. So we see a lot of these being living guidelines over time, and as the evidence continues to evolve, they will need to be updated regularly.
Thank you so much. Does anybody have any last questions for Dr. Edelmayer? If not, I do want to remind everyone this Friday, Friday the 18th, Good Friday, will be the Queen's Geriatric Conference at the Kroc Center. The focus is dementia, and Dr. Edelmayer will be one of our speakers at this conference. So we hope some of you can make your way there. Are there any last questions? I'll give a couple more seconds for people to speak up if you have questions. If not, thank you so much, Dr. Edelmayer. I apologize to you and our audience again for the mix-up in the Zoom link. Our staff is figuring out why that happened, and hopefully it won't happen again next month.
It's been a pleasure being with all of you. Thank you.
Thank you so much. Okay, bye-bye.